Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine a drug that acts like a tiny molecular eraser, specifically designed to just eliminate disease-causing proteins.
Powerful stuff.
Really powerful, right? But here's the counterintuitive twist. These drugs, they're called PROTACs, are often surprisingly large. And according to the usual rules, the sort of rules of thumb for drug permeability, they really shouldn't be able to get into our cells efficiently at all. So, how do they do it?
Yeah, that's the big question.
Today, we're diving deep into a groundbreaking paper from Cell, published in 2025, titled CD36, Gateway for PROTAC Drug Delivery. It seems to unravel this exact mystery. It identifies this unexpected gateway.
Right.
And it not only explains how these drugs get inside our cells, but it also seems to open up incredible new possibilities for, how we design medicines in the future.
You've absolutely hit on the core puzzle there. For decades, the rule of five has been a really major guiding principle in drug design.
Right, the Lipinski rules.
Exactly. And this rule basically suggested that for a drug to easily slip into a cell, it needs to be small and sort of greasy, technically, under 500 Daltons, so it can just passively diffuse across the cell membrane.
Okay.
Think of it like trying to get, a big sofa through a narrow doorway. Doesn't really work easily.
Yeah, makes sense.
But PROTACs, which stands for Proteolysis-Targeting Chimeras, they often way more, like over 800 Daltons.
Much bigger.
Much bigger. And they're designed to hijack the cell's own natural waste disposal system, the ubiquitin proteasome system, to specifically erase these disease-causing proteins. But despite their size, clinical data kept showing that some PROTACs were getting into cells surprisingly well. But, and this is key, with really unpredictable variability. Sometimes they worked, sometimes not so much. It was a real question mark for researchers, It strongly suggested something. Something far beyond simple passive diffusion had to be going on.
So, okay, the stage is set. Rule of five seems broken. PROTACs are defining expectations. How did these scientists even begin to crack this cellular safe? How did they find this elusive gateway?
Well, they started with a pretty clever technique. It's called target fishing.
Target fishing?
Yeah, imagine you're trying to find out what specific fish are in a lake, but you can't see them. You'd use a very specific bait.
Okay, yeah.
So in this case, they created what's called a biotin-tagged PROTAC probe. Basically, they attached a tiny molecular flag, the biotin, to the PROTAC molecule.
Look at a little label.
Exactly, like a label. They then used this flagged PROTAC to sort of pull down or capture any membrane proteins it stuck to on the cell surface.
Oh, I see.
After collecting these proteins that bound to the probe, they used a powerful analytical technique, mass spectrometry, to identify exactly what they'd caught. And one protein kept showing up, dominating the results, CD36.
CD36.
And that key step that's really laid out in Figure 1 of their paper.
CD36. Okay, that's fascinating. But was this a completely new protein, or was it something researchers kind of already knew about, maybe doing something else entirely?
That's a really pivotal question. And no, CD36 was definitely not new to science. It was primarily known as a scavenger receptor.
A scavenger receptor? What does that usually do?
Well, it was mostly recognized for its role in absorbing things like oxidized lipids and fatty acids, almost like a cellular sponge for fats.
Okay, so dealing with fats.
Right. But this study revealed its surprising, completely new role as a PROTAC gatekeeper. And they didn't just stop the mass spec finding. They went on to confirm this direct binding using another technique called surface plasmon resonance, or SPR.
SPR, right. That measures how tightly things stick together.
Precisely. It measures how strongly two molecules bind. And it showed that PROTACs, like one called ARV-110, bound to CD36 with incredibly high affinity. They reported a KD value of 5.9 nanomolar.
And lower KD means tighter binding, right?
Well, exactly. Lower KD, stronger bond. And to put that in perspective, this was much stronger than how CD36 binds to its known natural ligand palmitic acid.
Wow.
Yeah. So this indicated a really specific and robust interaction, not just some, random stickiness.
Okay, so they identified this potential gate, CD36, and showed PROTACs bind to it tightly. But how did they move from potential to proving it was the critical gateway? I mean, what happens if you, try to block that gateway or take it away?
Yeah, this is where the direct, you could almost say undeniable evidence came in. And it's shown really vividly in Figure 2 of the paper. They used genetic methods, specifically techniques called knockdown, basically to reduce the amount of CD36 protein in cancer cells.
So less CD36 protein available.
Exactly. And the results were, well, they were truly striking. PROTACs like Sim1Me and ARV110, which normally work quite well in these cells, were suddenly unable to effectively degrade their target proteins anymore. For instance, the actual concentration of ARV110 inside the cell just plummeted by almost 20-fold.
20-fold? That's huge.
It's a massive drop. And conversely, when they did the opposite, they knocked in or deliberately overexpressed CD36, making more of it.
Right.
The PROTACs worked much, much better. It showed a really clear dose-response effect. More CD36, better PROTAC action.
Okay, so it's directly related.
Directly related. And crucially, this wasn't just about the drugs getting inside. When CD36 was depleted, the actual anti-cancer activity of these PROTACs just diminished dramatically.
Right.
In some cases, they measured it as over a 400-fold decrease in potency.
Whoa, 400-fold.
Yeah. It powerfully demonstrated that CD36 wasn't just, helpful. It seemed absolutely essential for these specific PROTACs to function effectively.
That's incredibly clear evidence then. This obviously points towards a specific receptor-mediated pathway for PROTACs, meaning the cell isn't just passively letting them ooze in.
Exactly.
And this type of specific uptake, it's a known cellular process, isn't it? Like a sort of specialized internal delivery system the cell already has.
Precisely, yes. They showed that CD36 facilitates this uptake via a well-known process called clathrin-mediated endocytosis.
Okay, clathrin-mediated endocytosis. Can you break that down a bit?
Sure. Think of it like this. CD36 acts as the cell's front door, specifically recognizing and binding to the PROTAC. Once it's bound, the cell membrane kind of pinches inwards, forming a little bubble or vesicle around the PROTAC CD36 complex, pulling it inside the cell.
Ah, okay, like engulfing it.
Exactly. And this whole process involves specific helper proteins inside the cell, like ones called EEA1 and RAB5. You can think of them as the internal sorting and delivery crew.
Right.
They guide this vesicle to where it needs to go inside the cell. And crucially, when the researchers depleted these helper proteins, EEA1 and RAB5.
Let me guess, PROTAC efficacy dropped too.
You got it. PROTAC efficacy also significantly diminished. This further confirmed it's this specific active pathway. And, to really drive the point home, they compared it to a common chemotherapy drug, paclitaxel.
Okay.
Paclitaxel showed almost no dependence on CD36 for its entry or its activity, which really highlights how specialized this PROTAC delivery system truly is via CD36.
That's a great control experiment.
It is. And to visually confirm all of this, you can see in Figure 3 they used fluorescently tagged PROTACs, so they could actually watch these large molecules being actively ferried into the cell. And this movement depended on CD36. It really reinforced the whole idea of an active specific cellular delivery service.
That visualization is powerful. Okay, so finding this precise cellular delivery service for PROTACs is amazing. But does this gateway, this CD36 pathway, only apply to these specific protein-degrading drugs? Or could it be, a broader phenomenon in how cells take up larger things?
That's a really important question, and the answer turned out to be absolutely pivotal, I think, for drug development. This deep dive revealed that, yes, CD36 acts as a general pathway for other beyond rule of 5 or Bro 5 molecules, too.
Beyond rule of 5, right, so other big molecules.
Exactly. Other large and/or polar compounds, things like rapamycin, rapamycin, navitoclax, birinapant, tubicin, even doxorubicin, which is a pretty common chemo drug.
Yeah.
Some of these weigh over 2,000 Daltons.
Wow, much bigger than even the PROTACs they started with.
Much bigger. And Figures 4 and 5 in the paper lay this out clearly. So this discovery means it's not just some weird PROTAC-specific quirk. It fundamentally shifts our understanding of how many large molecule drugs might actually be entering cells. It seems to be a more general active mechanism.
Okay, so it's not just passive diffusion for many of these larger drugs. It's an active uptake system.
That's what the evidence strongly suggests. It breaks free from that old passive diffusion paradigm for a whole class of compounds.
So what does this all mean then for actual drug development? If CD36 is this crucial gateway, Can we, like, actually optimize drugs to use it even better, make them more potent, maybe?
This is where the innovation in the paper truly shines, and it's detailed around Figure 6. The researchers took this discovery and really ran with it. They actually started designing modifications to PROTACs, making prodrugs specifically engineered to be even better at binding to and utilizing CD36.
So tailoring the drug to the gateway.
Exactly. They added specific chemical handles to the PROTAC molecules. They made some more negatively charged, others more lipophilic or fat-loving, basically making them chemically more attractive to different parts of the CD36 receptor.
And how did those changes work?
Well, what's fascinating is how these modifications specifically interacted with the CD36 protein structure.
Yeah.
The negatively charged ones formed what chemists call salt bridges with a specific positively charged spot on CD36's surface, a residue called lysine 164.
Oh.
While the more lipophilic ones neatly docked into a key hydrophobic or water-fearing pocket on the receptor. So really targeted interactions.
Very precise. And did these tweaked procaccals actually perform better? I mean, did making them stickier to CD36 translate into better drug activity?
Remarkably so, yes. These optimized PROTACs showed significantly higher binding affinity to CD36. For example, one optimized version, called MZ1C12NB, had a KD of 2.8 nanomolar. Remember, lower is tighter.
Right, compared to the original MZ1's 9.6 nanomolar.
It's exactly. A much tighter bind. And this translated directly into much greater cell entry. We're talking up to 22-fold higher concentrations inside the cell.
22 times more drug getting in.
Yeah. And, as you'd hope, this led to drastically improved anti-tumor potency. Some of these optimized compounds were over 16-fold more effective in lab dishes, or in vitro, at degrading their target proteins. All this is shown really compellingly in Figure 7.
That is a huge leap in potency, just by optimizing for the CD36 gateway. And I heard something about it also solving a classic dilemma in drug design, some kind of trade-off it helped overcome.
Indeed it did. That's another really exciting part. Traditionally, medicinal chemists face this tough balancing act. If you make a drug more soluble in water, which is often good for formulation.
Like making it into a pill or an injection.
Right. But doing that often makes it harder for the drug to get across the cell membrane, reducing its permeability, and vice versa. Make it more membrane permeable, and its solubility often drops. It's a constant trade-off.
Okay, solubility versus permeability.
Exactly. But by specifically targeting this CD36-mediated endocytosis pathway, this active uptake system, they found a way to enhance both properties simultaneously.
Both? How?
Well, for instance, one of their optimized compounds, MZ1C14A, showed over a 1,300-fold increase in solubility.
1,300?
Yes.
Yeah.
While also boosting its cellular uptake by more than 20-fold compared to the parent compound.
Wow. That really is a game changer.
It really is. It sort of sidesteps the old trade-off by using this active transport mechanism. It proves that this chemical endocytic medicinal chemistry strategy, as they call it, can be effective across different types of PROTAC structures. It's like designing a highly soluble cargo that also comes with its own highly efficient delivery truck, namely CD36.
That's a great analogy. Okay, so lab results, chemical tweaks look fantastic, but the true test is always, in a living system, right? What did the animal models tell us about these optimized PROTACs?
Absolutely crucial question, and the results held up. In mouse models that had human cancer xenografts, so human tumors growing in the mice.
Right, a standard preclinical model. Right.
Exactly. In those models, the optimized PROTACs, like MZ1C14A and MZ1C12NB, were significantly more potent at inhibiting tumor growth compared to their parent unmodified compounds.
Better tumor shrinkage.
Yes, or slower growth. And importantly, this increased potency was achieved without increasing overall toxicity or negatively affecting the animal's body weight.
That's key. More effective, but not more toxic.
Precisely. This really validates the whole strategy for improving PROTAC's anti-tumor activity in a practical in vivo setting, which is, of course, the ultimate goal for any potential new drug.
This whole deep dive has really shown us a potential paradigm shift, hasn't it? Moving away from just size and greasiness to something more active. What are the broader implications, do you think, of this incredible work for how we approach drug discovery and maybe even patient treatment going forward?
I think this study fundamentally reorients our focus. Instead of just obsessing over passive permeability and strict molecular weight limits, it points towards the importance of what we might call the ligandability of uptake proteins. Yeah, essentially, how well can we design a drug to interact with and be actively escorted into the cell by a gateway protein like CD36? Think about it like how folate receptors are sometimes exploited in cancer imaging and therapy, using the receptor as a delivery route.
Ah, I see. So CD36 becomes another one of those targetable delivery systems.
Exactly. We now have CD36 as a potential new targetable delivery system for a whole class of beyond rule of 5 drugs, not just PROTACs.
So this could lead to entirely new ways to design drugs, maybe like adding a molecular zip code or something.
Absolutely. You could envision developing specific CD36 tropic delivery tags. Literally, a kind of molecular shipping label you attach to a drug to ensure it gets preferentially taken up by cells expressing CD36.
That could be huge for targeting certain tissues or tumors.
It could, and maybe even for getting drugs across challenging barriers, like, say, the blood-brain barrier for neurological diseases, if the right cells there express CD36. And this also has really profound implications for precision medicine.
Well, the study showed there was significant variability in how much CD36 protein was expressed across different prostate cancer tissue samples.
So some tumors have lots of the gateway, some have little.
Exactly. This means we might eventually be able to screen patients, perhaps biopsy their tumor, check their CD36 levels, and then stratify them. You'd give these CD36-dependent drugs only to those patients whose tumors have high CD36 levels, making them most likely to benefit.
Increasing response rates and avoiding side effects for others.
Right, potentially increasing response rates and reducing unnecessary side effects and costs for patients whose tumors wouldn't respond well anyway. It's also worth mentioning the relatively low hematologic toxicity often seen with PROTACs, meaning they don't hit blood-forming cells too hard.
Which is a common problem with chemo.
Big problem. Well, this low toxicity might be partly explained by potentially low CD36 expression in our blood stem cells, so the gateway isn't really open there.
That's another potential benefit. Okay, this all sounds incredibly promising. What about remaining challenges or, the really exciting future directions? Where does the research go from here?
Well, there's always more to learn, of course. For example, one interesting point is that CD36 is also highly expressed on immune cells called macrophages. Theoretically, these could act like a sink, soaking up the drug before it gets to the tumor.
Okay, so potentially diverting the drug.
Potentially. But current PROTACs actually show surprisingly low toxicity in other CD36-rich tissues, like the gut and the liver.
Yeah.
So it's not straightforward. It suggests there might be nuances in how CD36 functions in different cell types. Maybe it depends on subtle chemical modifications on the protein or different downstream processing pathways inside the cell.
So more biology to unravel there.
Definitely. And future studies will undoubtedly explore whether other scavenger receptors, besides CD36, could also ferry different types of chemically distinct degraders or large molecules into cells. Are there other gateways we don't know about? But ultimately, I think this discovery really lights up exciting new pathways. Pathways for potentially improving the oral bioavailability of large drugs, tackling those really challenging drug delivery barriers, and maybe even combining PROTACs with other novel approaches like lysosome-targeting chimeras or LITACs for even more comprehensive protein erasure within the cell.
What an absolutely incredible journey from, a puzzling observation about large drugs getting into cells when they shouldn't, all the way to uncovering this fundamental cellular gateway, CD36. This deep dive really reminds us that even in fields we think are well understood, there are always these unexpected biological mechanisms just waiting to be discovered.
Absolutely.
Mechanisms ready to potentially revolutionize medicine. It just goes to show you that sometimes the rules in biology, like the rule of 5, are maybe more like guidelines, ready to be reevaluated.
Indeed. And I think it leaves us, and hopefully you listening, with this thought. As we uncover more about these intricate cellular delivery systems, these gateways like CD36, and as we learn how to chemically talk to them more effectively, how might our understanding of these pathways really transform how we approach treating not just cancer, but maybe a whole host of other diseases where getting the right drug to the right place remains a major challenge?
Definitely food for thought. Well, that's all the time we have for this deep dive. We hope you feel a little more well-informed and maybe inspired to explore this exciting world of receptor-mediated drug delivery on your own. See you next time.